Hey everyone, I wanted to talk to you a little bit about microbial genetics. So I'm going to go through these slides fairly quickly, but you will have them. You can bring questions to our Zoom meeting and we can talk about them more, but I wanted to make a short recording and point out some of the highlights. So here on this slide where we have hereditary material, all organisms have genetic material. And that is in the form of chromosomes. Chromosomes are made up of DNA, and DNA, it's a kind of molecule called the nucleic acid. DNA has these little sections on the chromosome of genes. And each gene you can think of like a recipe. So if the chromosome was a recipe book, and you flip through the pages to see what does the cell want to make, what do you want to make for dinner. If you flip through the pages or you look through the chromosome, within it there are recipes or genes, and the genes give instructions to the cell for how to make things. And most of the things that it makes are proteins, but there are other things that don't code for a protein. So a genome is the total of all genetic material that an organism has. So it's going to include the chromosome, but there's other kinds of things that it can have. So many bacterial cells, for instance, have something called a plasmid. Where's the plasmid? It's over here. And this is extra little pieces of genetic material that can be useful for something. So maybe it has a gene for antibiotic resistance. Maybe it has instructions, a little recipe for how to digest some kind of nutrient like lactose. Normally the cell doesn't need to do that because there normally isn't lactose in the environment. So that might not be in the chromosome, but there could be a plasmid that has specific information about that, for instance. So what else is in the genome besides the chromosome? If that cell is eukaryotic, then it will have mitochondria. It will have, if it's able to do photosynthesis, it may have chloroplast. So depending on what the cell has, there would be other genetic material inside there as well. So genome means all the types of genetic material, basically all the DNA, and if it has RNA, like in the case of a virus, then that would be considered its genetic material, its genome. So genomes vary in size, and here we're mostly looking at we're thinking mostly of chromosomes, but any genes that are in there. So for a virus, it will just have a few genes. For a bacterial cell, it may have around 4,000. That's what E. coli has. For human cells, like ours, we have about 25,000 genes. So you can think of it as recipes. So the ability to make or do something with that genetic information. All right, let's talk about something called the central dogma of biology. And so this overall principle, which more or less works, there's some exceptions, but this overall principle is the idea that the genetic material inside DNA gets copied and then it can be used to make protein. So there's three specific terms that I want you to be familiar with. I know some of you have taken biology, and so you probably already know about this, but if you don't, or if it's been a while, then I want you to review it. So one of these processes is called replication. So when a cell copies its DNA, so we can think of that as going from DNA making a copy of DNA, that is called replication. When would a cell want to do that? It would want to do that before the cell was going to divide. So you can picture a situation where a cell wants to divide. It needs to copy all its genetic material so that each new cell can have a copy. So that's what replication is, and we're going to talk about that some more. That's one of the main focuses of this little lecture. Transcription is what happens when that cell wants to make a copy of just a gene. So what do you want to make for dinner tonight? You might go to the recipe book and pick out something specific, chicken noodle soup maybe. Well, in the cell, it might need to make something specific. Maybe there's a particular enzyme it needs. Maybe it needs to make something for the cell membrane. Whatever it is, it could do that through transcription. So in transcription, a copy of the DNA gene is put into a temporary form, and that is into RNA. That RNA can then be turned into a polypeptide protein. And so when we take that RNA and turn it into protein, we call that translation. And so it's kind of this three-step process from DNA to messenger RNA. That's the type. There's different kinds of RNA that can be made. And then in the ribosome, it gets translated into a protein. But everything starts with the genetic material, the recipe that is inside DNA. All right, so in talking about DNA replication, there are some important structures, there are some important enzymes that are involved, and in the module there's some good information about there, but let me just briefly go over them. So how do we know, how does the cell know where to start copying, where to begin DNA replication? Should it begin on the side, on the bottom? Where does it begin? So the ORI site, or origin of replication, tells the special enzymes that are going to do the replication where to start working, where the beginning is. Helicase, it's what's going to open the DNA up. So we're going to look at the structure of DNA, but helicase is the one that opens it so that replication can get started. Primase. So this is an enzyme that is going to make a little temporary RNA primer. So it's going to put little nucleotides of RNA on the ORI site. And that happens at the beginning of where this replication is going to happen. So we will look at another picture that'll highlight that a little bit better. But the RNA is kind of a temporary form, and we'll talk a little bit about why. DNA polymerase 3, this enzyme, anytime you see ACE, that's an enzyme. This enzyme is going to add new DNA nucleotides when we're copying that strand. And so here we open up and we want to copy the strand. And so this is the enzyme that can add the new nucleotides. So the A's, the T's, the G's, the C's. DNA polymerase 1, this is an enzyme that is going to remove the RNA primer that primase put there, and it's going to replace it with DNA. And at the end of the process, ligase is going to seal the DNA back up. There'll be little holes, and it will seal everything like glue. All right, let's take a look at this DNA molecule. And so I want you to notice some things. First, there's two strands. So DNA is really in a helix, kind of twists around each other. But if we straightened it up, we would see that there's two strands, and that in the center they are connected through these nitrogen bases. So this A adenine always goes with T thymine. This C cytosine always goes with G guanine. And so if we know that there's a C here, for instance, it always will pair up with a G. If we know that there's a G, it always will pair up with a C. Another thing I want you to notice is that there is a 5' end, and at the bottom, it's a 3' end. And notice that the opposite strand is the opposite. So notice that this is 3, and we can't maybe see the bottom down here, but at the bottom, there is a 5 down there. So that direction has to do with the sugars, the carbons on this sugar. And so everything is labeled, and so we call it prime. So that this ends up referring to where these molecules attach. And so that becomes important because we are always adding a new nucleotide to the 3' end. And what that means is that the strand is growing from the 5 to the 3 direction, because we started with the 5 and now we're continuing in this way. So the direction is from 5 to 3. And so that's always important. We add to the 3' end, and the direction is going from 5 to 3. So we know which nucleotide we're going to add. It has to be a T, because on the template strand here, there's an A. And it takes the energy in these phosphate bonds. It's going to break off there, and it's going to leave one of the phosphates, and then 2 of them are released. And so you'll notice that this is called a sugar phosphate backbone. So here is a phosphate, it's a little circle here, and then there's a sugar, and then phosphate, sugar, and it continues all the way down. And so continuing on, what would be the next one? Well, we would add a G over here to this C. And so that's how that works. All right, I put this slide here just so that we can look at the nucleotide a little more closely. So the nucleotides that make up DNA, and RNA for that matter, have 3 parts. And so what do they have? You can see up here in the upper right corner, they have a nitrogen base, a 5-carbon sugar, and a phosphate. So the nitrogen bases, here they are here, so the cytosine, thymine, adenine, and guanine, those are the 4 for DNA. They pair with each other, and here you can see some of the numbering on this, on the sugar. This is also another kind of good picture here. So I wanted you to see that this is the kind of sugar in DNA. It's called deoxyribose, and RNA, it's ribose. They're very, very similar. Just this is the only difference, the hydrogen and the hydroxide at the bottom there. It's the only difference. All right, let's take a look and briefly go over DNA replication. And this is something that we can look at in the Zoom meeting and see if anybody has any questions about it. But basically, actually, what I want you to do is to spend a little time and read each of these sections. So it's numbered here, and it's showing you what exactly is happening. What's also great is that down here at the bottom, it has a little key, so you can see what these things are. So here's our double-stranded DNA, and it is being opened up, and here by number 1, you can see that is the helicase. So that helicase enzyme is opening up the double-stranded DNA. And then on one strand, which is called the leading strand, and don't worry if it happens to be on the bottom or top. It just depends on the drawing. But here's the leading strand. And remember that DNA is always synthesized. All nucleic acids are, by the way, always synthesized, remember, in the 5 to 3 direction. So we add to the 3' end, but the overall direction is 5 to 3. Okay, so what has to happen is that before DNA polymerase 3 can start synthesizing the DNA, so for this whole replication, the synthesis to begin, before that can happen, we have to add a little bit of RNA primer, and you can see that in green here. So here's the ORI site, origin of replication. That's where the opening happened. So it opened up. There was a little bit of RNA primer added, and the enzyme that does that is the primase. So it adds a little bit of RNA, and then DNA polymerase 3 starts synthesizing, meaning it adds nucleotides, and that continues. This is called the leading strand. The other strand is called the lagging strand, and synthesis is going to happen in the opposite direction, and it's going to be in little pieces. And that has to do with DNA polymerase 3. So there's two issues. One, it can only synthesize in the 5 to 3 direction. That's one thing. And so if you take a look at this, in order for it to go, look at the top strand, in order for it to go from 5 to 3, it has to go to the right. Notice the bottom strand is going to the left. So synthesis has to happen on this top strand, moving to the right, and that means that DNA has to open more and more, and each time, this is called the fork, each time this replication fork opens, then we can synthesize another little piece. So we end up getting all of these little fragments, and they're called Okazaki fragments, after the Japanese scientists that discovered them. So then next what happens is DNA polymerase 1, shown here in this purple. It comes along and it has to remove the little RNA primer that primase added, and it replaces with DNA. And then at the end, here's the little ligase. There's little tiny pieces where the DNA is not connected all the way, and so it's going to seal it up, and that's the ligase. Now, why do we have to add RNA here? Why not just DNA? And the reason, so DNA polymerase 3 has to have a little piece of double-stranded molecule to start. So in other words, it can't just come if there's a single strand by itself and just start adding nucleotides. It has to have a little piece on the end. It's kind of like a zipper. If you can imagine trying to zip something up, at the end of the zipper, you'll notice it's a little bit different, a little bit smoother to grab on. And so that's kind of how DNA polymerase 3 is. If you just had single-stranded DNA, it can't start the synthesis process. So we add a little bit of RNA nucleotides there, and then DNA polymerase 3 can attach, and then it starts zipping. Then it starts adding the nucleotides. But then we have to come back and say, well, we want the finished product to only be DNA, so we have to remove that little bit of RNA. All right, transcription. And so let me flip back a little bit. Let's just remind ourselves. Let's go back to this central dogma page. So we started with replication. Remember transcription. If we want to use one of these genes, if we want to make something, we are going to have to transcribe it. We are going to have to make an RNA copy. And that temporary RNA copy is what gets turned into protein. So we don't directly use the DNA. We make it into RNA and then we turn that into protein. All right. And so transcribing DNA, making a copy of just the specific gene or genes that we want to use, right? You don't go home and cook every single thing that you have in your refrigerator and your cupboard and dump it all in one pan. You make and take out just those ingredients that you need for dinner tonight. And that's what the cell does too. It doesn't copy all of the DNA because the cell's not dividing all the time. And so for transcription, it's just only going to make copies of those genes that it needs right now. And so we make RNA. And there's different kinds of RNA, but the one we've been talking about is messenger RNA. And that messenger RNA is going to go to the ribosomes, and prokaryotes and eukaryotes have ribosomes, and then that is where it will get turned into protein. Another kind of RNA is ribosomal RNA, and that's part of what makes up a ribosome. Also has protein. And then the tRNA, transfer RNA, that's involved in bringing an amino acid to the ribosome as part of translation. But we're not going to cover translation in this lecture. But translation is that third step where we finally make our protein. All right, in looking at transcription, there are a few steps that I want you to know. So one thing is I want you to notice that we use RNA polymerase. Remember before we were using DNA polymerase to make DNA, but for making an RNA molecule, we're using RNA polymerase. And this molecule is DNA because what we're going to do is make a copy of the gene that's on the chromosome. So we're copying the DNA. The beginning of the gene we can find by the promoter. So that's the region of DNA right before the gene. It's telling RNA polymerase where to get started. How does it know where to end? Well, there's a termination sequence at the end, and the RNA polymerase will fall off, and then it knows it's done. Here is the little RNA transcript that is being made. So we're making a temporary single-stranded RNA copy of the DNA. This is showing you the relationship between the DNA, the RNA, and protein. So the DNA, 3 triplets, so this would be 3 nucleotides, are going to correspond on RNA. So 3 nucleotides make up a codon, and then that will correspond to a specific amino acid. This is how you get the protein in the end. So this is sort of the language. This is the language in the DNA, the double-stranded. It's a single-stranded RNA that's complementary to one of these strands, not both, to one of them. That's going to be the template strand. And then ultimately this gives us a code for which amino acid to add. All right, let's talk a little about genetic engineering. And so scientists learned about all of these enzymes and how they work in the cell, and they've been able to use them in the lab to kind of manipulate and work with DNA in a test tube. So outside of a cell. And so those same enzymes that we were talking about before. 3 of them are here: helicase, ligase, and DNA polymerase. But there's a couple other ones that I want to mention. One is reverse transcriptase. And that is a very special enzyme, and we know about that because some RNA viruses have that, and that allows them to go in the reverse. Instead of going from DNA to RNA, they can go from RNA to DNA. And then another special one is called restriction endonuclease, and this is going to allow us to cut DNA in special places. So it was discovered that some bacteria had these special enzymes that allowed them to protect themselves by cutting up this foreign DNA from viruses. So if a bacterium was infected by a virus, it could cut up the viral DNA using these endonucleases. Well, scientists discovered, hey, these are great tools, and so we can use them to cut DNA ourselves, and it's a very useful technique. All right, so I don't want to spend a lot of time going through this, but I want to show you. So at the top, you can see this long piece of DNA, and I'll let you read through this and take a look at it. So here's the DNA. We're going to get an RNA transcript, and it actually has to be processed because there's pieces of the DNA, introns, that are not needed. So those are removed, and only the exons make up the final product here. And then look at this. We can use reverse transcriptase, and we can take that RNA and we can make a copy, and then we can do it again and now get a second copy. So it's double-stranded now using just the regular DNA polymerase. All right, so let's just take a look at how these restriction endonucleases work. So they're enzymes that cut DNA, and they cut them at very specific places, and those are called palindromes. And so each enzyme is different, and you can buy them for $30. There's catalogs. There's probably 1,500, maybe more, that people have discovered, and they're named after the bacteria. So there's one, EcoR1. Guess who it's named after? E. coli, because it was discovered in E. coli. And so this was a tool that this bacterium used, and now we can use it to also cut DNA. And so they figured out exactly where they cut the DNA. And so if you know what the sequence of DNA is, you can specifically choose the enzyme that will cut it exactly right there. All right, want to talk a little bit about some of these techniques. So gel electrophoresis is something really interesting. We can take a sample of DNA and we can cut it with these enzymes, and in this process we actually put the DNA in these little wells. We put a buffer over it. There is a little electric charge here, so we run a current through it, and then the DNA will migrate, and DNA happens to be negative, so it's going to be attracted to the positive well, and so it'll migrate through the well, and then based on how far it moved, it will make patterns. You can stain it. There's different dyes that you can use, and you can compare DNA. And so let's take a look. So why would this be useful? Well, everybody's DNA is different. And one reason they're different is that we have different sections of these introns, and there's random mutations. And so everybody is unique, like a fingerprint. It's referred to as polymorphisms. And so these different combinations of different sequences make us sort of who we are. in terms of being unique with our fingerprint. And so the more closely related you are to someone, the more that you will share. People from similar regions will share some similar polymorphisms. You're going to get half of them from each parent. So let's take a look at an actual sample. So using DNA fingerprinting, so taking this DNA and you use enzymes to break it up. If you just ran the whole DNA, it's going to be too long, very big. So you're going to break it up and then run it through these gels. And so taking a look at this, we put something called a marker or a ladder. So in this bottom image, you can see there's 3 markers there. And so these have pieces that are like controls. You know exactly how far they're going to migrate. And then you can put samples in there. And so in this sample, we have some evidence, we have some suspects, and you can compare them. And so they could say, okay, this is the evidence found, at the crime scene, and does that match either of these suspects? So you can see that it matches suspect number 1, for instance. And so that gives you this extra piece of information. Hybridization, and so you can form a connection between 2 DNA strands or a DNA and RNA strand, and because of the way DNA perfectly matches up the A's and T's and the G's and C's, or if we were using RNA, then it would include uracil, and that would bind with adenine. So because it always is complementary and binds exactly, there are different diagnostic tests that can use these. So if you're looking, let's say, for a specific microbe, then you could have a DNA that will exactly match it. And if it binds and matches, then it could release a color. And then it would be positive identification that you'd say, oh, this is salmonella or some other organism. It would give you a positive ID because it's very specific if the DNA exactly matches that sequence. And so here, DNA analysis used in the same way. All right, 2 more things to talk about. PCR, polymerase chain reaction. And so Kary Mullis came up with this, the scientist, and he got the Nobel Prize for it, but figured out a way to make copies of DNA. So if you just had a little tiny sample, let's say that you found like an old fossil or something and you wanted to study it, but you don't want to use it all up in studying it, you could make many samples of the DNA. Or same thing, maybe from a crime scene, you just have a little bit of evidence, you can make more copies of the DNA. So it's kind of like a copy machine. And so you have 1 piece and then you turn it into 2 and then 4 and then 8, and you keep copying it on and on. So this slide shows you the basic steps, and you can see that it's going to repeat. So here's 1 cycle at the top, and then they're showing 2 sets, and then that's going to be repeated. And so each of these keep doubling, doubling, doubling. And these are the 3 main steps. So first you're going to have to heat it up. That's denaturation in order to split the DNA. Now, if it's in our cell, we don't need that high temperature. But if you're trying to do this in a test tube, then you need to do it. And so we heat it up to 94 degrees to separate the strands. Then we have to add, remember our little primer, our little RNA primer. And so we cool it down to 50 while we add our primer. And then we need a DNA polymerase that can synthesize the new strand, and that's called extension. This is done at a high heat. Our body temperature is 37, And so you notice 72 degrees, that's way hotter. But this is a very special kind of DNA polymerase, not the ones in our cells, but it's another one that's used to this very hot temperature. And so it can extend and synthesize the new strand there. And so we now have 2 copies, and then we'll have 4 and 8, and it'll keep doubling like that. All right, this is the last slide. This is gene therapy, and in the module I put a link to a couple good resources there. But the idea is that you are going to either replace or repair a gene. And so very often this is done by taking stem cells out and then repairing the gene and then putting that back into the person's body. And so if it's ex vivo, that would be the example that I was just talking about. If it's in vivo, then you actually would make the change in the person's body. So you would try to repair the gene in their body. Sometimes viruses are used, and so these are good viruses, helper viruses, that are used to try to get genetic material into the cell while it's in the person's body. That would be in vivo, and it is risky. And then ex vivo is the first one that I mentioned. So take a look at that. I want you to read about gene therapy and DNA fingerprinting and PCR, those 3 especially, and then we can talk about it some more at our Zoom session. Okay, see you all soon.